Okay, thanks, Pam. Thanks everyone for being here, and a big thanks to Fran for accepting our invitation to give a seminar here. So a little bit about Fran. I had the pleasure of meeting her in person, even though we had some personnel overlap over the years in Tokyo, actually, right before COVID, that was. And then we've kept in touch over the years. So Fran is a professor at Memorial University of Newfoundland, where she works on sustainable catalysis, some related to the ocean, some related to CO2, which is again related to the ocean. Most importantly, she's an associate editor of RSC Sustainability, and she currently chairs the IUPAC. So for the non-chemists, that IUPAC stands for International Union of Pure and Applied Chemistry. So basically, if you're making a new molecule and you want to name your molecule, you have to touch base with IUPAC to make sure you're abiding by their rules. So Fran got her PhD from the University of Sussex in the UK, and then I believe moved to Canada in the very British part of Canada called British Columbia. And I do note that she ultimately found a home in Newfoundland. So there's a definite British trend there going on. Anyway, so most importantly, she's working there on developing catalytic systems related to sustainability, and is now heading the NSERC-funded Center for Innovation and Research on Carbon Dioxide Utilization. So without further ado, I'll hand over the floor to Fran.
Thank you for the kind introduction, and also thank you all for the opportunity and listening to what I have to say. I'm very happy to answer questions at the end. I know that there'll be some aspects that are more interesting to some of you than others, and also some parts that might be more confusing than others, but I've tried to keep it quite kind of general as to what we do. So within my group, as Shrin implied, our research is centered on catalysis, and we do a lot of materials chemistry. And within that area, we use a lot of biomass feedstock. So I'll be talking about some that are derived from waste from the forestry industry, but a lot of what we do is waste from the seafood industry. And we've started doing a little bit of work with seaweed as a feedstock. We do quite a lot with carbon dioxide utilization, so trying to add value to CO2 and also that way have it stored as a bit of a carbon sink. And then also we've been looking at biodegradable polymers and materials that we can access from biomass. So the micrograph at the top left of the screen is of calcite crystals end-on that are present in the outermost part of mussel shells. So a few years ago, I did a word cloud. I need to do another one and update it, because I always used to say, oh, my research is centered on catalysis, but then was it really? And so catalysis or catalyze was a top word in the abstracts and papers that I've been publishing. So I wasn't lying about that. So I was probably one of the first people in Canada to sort of go by the label of being a green chemist. So chemistry has a pretty bad rap worldwide as sort of polluting the environment, but actually chemists are important in providing the solutions to stop pollution happening. And so 25 years ago, last year, Anastas and Warner came up with the 12 principles of green chemistry that are kind of a philosophy or a tool for determining whether your process or anything that you're doing with chemicals is green. And so the ones with the check marks on it, these are the ones that we really like to use in my research group. And to be honest, number 12 happens by chance if you employ as many of the others as possible. But a key one that we'll be looking at today really is the use of renewable feedstocks. And so that's number 7. And so renewable feedstocks, we can get them from any form of biomass. A lot of the focus has been on plant-derived biomass. And so, for example, soybeans, you can get a soybean oil, which contains different fatty acids that can be used for making surfactants, so soaps and things. That's quite useful. One of the most commonly used biodegradable polymers is made using lactide. It's called polylactide or polylactic acid. It's used in 3D printers and all sorts of places. And that lactide is obtained basically from the starches in crops like corn. However, there are downsides to using renewable feedstocks like this, in that you're competing with food, and also, you'd be much better off using waste. And the land used is obviously competition with habitats. And so I'm one of the people in the world who's been looking a little bit more at ocean-based renewable feedstocks. And for example, in shrimp and crab and lobster shells, there are there's one biopolymer present, but it's called chitin, which has the structure shown here. It's not that important to know its structure, but just to know that you can do chemistry with these biopolymers and change their structure. So we can change the blue structure of chitin by removing the oxygen-containing group attached to the nitrogen, and produce chitosan that has a number of different uses. And by using green chemistry, we can address the United Nations Sustainable Development Goals. So I just thought I would highlight them. And for example, you've got number 2 here, zero hunger. So chemists work a lot in that area in terms of helping improve agriculture through agrochemicals. But also we've got to consider, if we're using something like corn as a feedstock, that's going to affect zero hunger. So we want to avoid using our food biomass. We want to use waste biomass wherever possible. But we can also impact climate change, life below water, so many different sustainable development goals. And it's becoming more and more kind of common as a indicator that when you write for funding, that they're asking you which sustainable development goals are you really trying to address. So I'm going to talk about 3 of the projects in my group. The first one I'll talk about is basically trying to understand the material biochar more, and that's done in collaboration with Dr. Stephanie Macquarie of Cape Breton University. Some of the materials that we've made from waste ocean biomass, particularly focused on mussel shells, and then finally a little bit on CO2 derived polymers. And I hope I won't have lost you by then. So this is a little cycle, the kind of simplified version of the carbon cycle of relevance to forestry. And so obviously we're capturing CO2 while trees are growing, and then the trees are harvested. But there are waste materials from the industry, such as barks and twigs and things that aren't used in making, for example, furniture and building materials. And so normally these are left on the forest floor to decompose, and they produce CO2 and methane. But the Intergovernmental Panel on Climate Change actually highlighted in their report that biochar could be a way to trap that CO2 that's been sequestered in plants. So this is done by pyrolysis, and it's actually a very ancient method used by lots of cultures in South America to make fertilizers historically. But basically you heat up in the absence of air, you produce some oil and gas that can be used as a fuel to help in the pyrolysis, and then you end up with this carbon rich material where the carbon is trapped, so you're not producing as much CO2. So Dr. Juliana Vidal, who completed her PhD in my group in 2021, worked on this project and published a couple of papers on using biochar in catalysis, use of biochar in making degradable polymer composites, and also on some exfoliation to make nanostructures. And she started a collaboration with the University of Florida and lots of other activities. And she works for an educational foundation now, still based in Canada, but that foundation is based in the USA. So what she was really trying to answer was, some of you may have heard of the material graphene. It won the Physics Nobel Prize a number of years ago. And she was trying to see, we make this carbon-rich material. Can we consider it to be similar to graphene or graphene oxide, which is graphene with some oxygen groups on it? And so in 2004, the Nobel Prize winners in physics, they used Scotch tape to break apart the layered structure in graphite to make single 2D layers of graphene. And these thin kind of mono layers, they found they had very exciting properties. And later it was found that you could scale this up a little bit more easily if you used ultrasound. And basically, with a solvent present, you can break the interactions between the layers in these layered materials and produce the same sorts of nanostructures that they'd originally made using Scotch tape. And what people found is that these sorts of processes, this liquid phase exfoliation, depends a lot on the solvent properties, so the density of the solvent, but also the surface tension. And some of the best solvents for this process in terms of how much they could break apart were highly toxic. So that's where the kind of green chemists came in. And in our group, where we're interested in green chemistry, when we were exfoliating biochar, we wanted to use green solvents. So we went through the literature and made a big table of green solvents to try. We also noted down their density and surface tension, but also some other properties that are more chemistry related. So this pi star is how many electrons there are in clouds around the molecules of solvent, and therefore if you have high values of this, they should be able to interact well with the graphene-like portions of biochar. And so the scales for this pi star is 0 to 1. And what we found was if you had a higher than 0.5, so 0.8 or something on this 0 to 1 scale, it was able to disperse the biochar better than those with low values. And so we had a number of solvents that were less dangerous than the ones typically used. And so I'll draw your attention to acetone at the bottom right. Acetone is typically used as nail polish. It's something that we can produce in our bodies sometimes, and so it's generally regarded as fairly safe. We also looked at a material called oxidized biochar, where we've introduced OH and carboxyl groups on the surface, and we found that for this, the solvent parameter, this Camelot-Taft parameter that was important, was called beta, which means hydrogen bond accepting ability. So all our solvents that had oxygens in there that could form hydrogen bonds with the OHs on our biochar, they were good at dispersing that. And so this included things like polyethylene glycols, which means they're non-volatile solvents, so they have very low flammability, and a molecule called epsilon caprolactone, shown on the right here, that I'll talk a bit more about, but it can be used to make degradable polymers. One of the things the referees wanted to see when we submitted this for publication was if we increase the time, did we make it that there was less graphene-like portions in our material? And so this ratio on the bottom bar chart shows you that over time, the ratio that we're measuring here, which if there was less graphene would go lower on the scale, would be down at 0.2, stays about the same. And if we increase time, we can get more material dispersed in our green solvents. And so we did lots of other analysis that I'm not going to bore you with, but using microscopy, so atomic force microscopy, we could figure out how well we'd managed to separate the layers and so on. And so when Juliana found that she could disperse the biochar, represented by the little smiley faces, within this caprolactone material that can act as a monomer, she was very excited and says, okay, can we use it to make some degradable polymers that have a little bit of stored carbon in there in the form of this biochar. So she would exfoliate to make the dispersed biochar, and then she used different catalysts, including enzymes, to make polycaprolactone with the biochar trapped in it. And she was able to find that the biochar, if we'd exfoliated, really didn't have a significant effect on the molecular weight of the polymer. Whereas if we didn't exfoliate it, it had no effect. Whereas we got a lower molecular weight if we exfoliated it. But what we found was with the exfoliated biochar, we got a much more stiff composite. Hopefully the little animated GIFs are working, and it was much more crystalline than neat polycaprolactone. So we showed that it makes a difference, but in actual fact, the actual mechanical properties weren't better than what we had before. For many applications, you want some of the flexibility still in there. And on the right, one of the things we've started doing is trying to look at degradation, both mechanical, biological, and chemical degradation of the polymers that we're making. So in this case, it was just a mechanical process. But what you can see on the SEMs is that by putting the films in water, you started to see a lot more cracks. And part of this is that the biochar produces some kind of defects and sites for the water to permeate into the material. And we think that this might also help in terms of biodegradation studies. One thing I learned through working in this area is don't always believe everything you read and what you're told on a superficial level, because some of the so-called degradable polymers that are sold and promoted, they're actually take a very, very long time to degrade in the environment or in your typical landfill site. And so it's just something to be conscious of. So the final bit of story of the biochar is actually where I started my interest with biochar. So we got this big chicken wire type structures here. These are just meant to represent simplified forms of biochar. And so when you pyrolyze your cellulose and other materials in the tree waste, it gets rid of a lot of the oxygen in the form of bio oils and so on, but we're also producing still got some remaining oxygen containing groups. And so what Stephanie Macquarie's group discovered was if you actually intentionally increase the number of oxygen containing groups, so these are the orange groups on the middle structure, you can then add functional groups, including amines, to the surface that's shown in red. And the reason she was doing this was to make an adsorbent for trapping CO2 with the amine. And when she was talking about this, I knew that amines like this had also been used in some catalytic conversion of CO2. So I said, oh, can we collaborate and try that? And so Ju's first project when she joined my group was to look at this reaction on the right of the slide, where we have a 3-membered ring molecule with oxygen and CO2. We can put them together with our, in this case, oxidized biochar and make a 5-membered ring molecule. And this molecule is called a cyclic carbonate. And there are lots of interesting uses. Perhaps the most important in terms of sustainability is that they're used in the electrolytes for batteries and other electrochemical applications for energy storage. So we're quite pleased with this work in that we could use a form of stored CO2 and carbon in our oxidized biochar, but then even use that as a catalyst to trap even more CO2 in the form of these carbonate molecules. So the first catalyst we tested was the amine compound that Stephanie Macquarie's group had made, and we were like, that's going to be the best, and it was in fact the worst. And then we used biochar straight from the reactor where Stephanie makes it, and that could give us a little bit of conversion, 40% for our model compound that we start with. And then if we oxidize the biochar, it increased not quite twofold, but quite a lot. Juliana then changed the reaction conditions and found the best temperatures and pressures and time and so on, because in this sort of chemistry, we're wanting to try and reduce the pressure. So ideally, we want 1 bar or atmosphere of CO2. We'd rather have room temperature and shorter reaction times, but this is pretty good considering this is a catalyst made from a waste material from forestry. And then we can test to check how well it works. And for most of these 3-membered ring compounds, it worked quite well. And then we could also reuse it many times and not see a reduction in its ability to make the new product that we wanted. So I'm going to change a little bit now and talk about some of our work with ocean biomass, which is a bit different from what other people are doing in Canada. And part of the reason for doing this was just to try and take advantage of the geographic location of Newfoundland when we came here. And at this point, I also like to promote that it's the United Nations Decade of Ocean Science for Sustainable Development. So there's lots of exciting research going on around the world in ocean science. And that might be from a physics perspective or a geology perspective or a biology perspective. There's many different people working in that area because, of course, our planet is, 70% or more oceans. So in Newfoundland, the aquaculture industry has been growing a lot over the years that I've been living here, which is 18 years now. And it's been growing a lot worldwide. If you Google United Nations aquaculture growth, you'll find lots of reports from them. And there are challenges involved with that. And so people are developing new ways of making fish feed, so artificial fish feed or fish feed that doesn't rely on wild caught carp fish, and other people working on biopolymers that are produced from aquaculture waste. But it's not all positive. I mean, it's good that we can address maybe Sustainable Development Goal 2, which is zero hunger, but there are problems with pens like this that are out in open water that fish can escape or catch disease and contaminate the environment. I've just got one slide on this project, which is a new project that was started in fall 2022 and is a collaboration with the National Research Council of Canada. And this is on fish bones. So this is Sarah Boudreau's project in my group. And what we've done is we've used enzymes as environmentally friendly catalysts to basically clean the meat and other material from fish bones. And what was great working with the NRC was we were able to scale that up. So we could isolate 120 grams of hydroxyapatite from 15 salmon frames, whereas in our lab, the most we could probably do is about 1 frame to give us 10 grams. And so that's been really helpful as Sarah moves forward to try and find interesting new uses for this material. Our first work, though, with ocean biomass focused on mussel shells, and this was mainly because I was a bit afraid of going into the biological arena at first when I started work here. And so I'd spoken with the Newfoundland Aquaculture Industry Association, and they said 90% of what we do is fin fish farming, like salmon or trout. But we also have quite a lot of mussels. So I was like, okay, I'm going to do something with mussel shells. So the problem with mussel waste is that you can either have dead mussels, which obviously have the decaying organism in them, or you can have the residual protein or the abductor mussel stuck to the shell. And so if you store this waste so that you have enough to actually make it worthwhile using, it starts to smell. It doesn't smell good. Lots of kind of ammoniacal smells coming off it. So people are interested in removing this protein. In the literature, people had used heat to very high temperatures, over 500 degrees C, or they'd used jets of high-powered water, and that's a valuable resource that many rural areas wouldn't have, or they used wire wool and abrasion to remove the protein by hand. So the Newfoundland Aquaculture Industry Association, DuPont had been in touch with them and said, oh, why don't you try using enzymes? So together with us, we developed a enzymatic process where we could remove the meat or meat residues and produce a biomineral stream. And the enzymes are really cool because on the packet it said, oh, you need to use these at the optimum temperature of 60 degrees. They worked perfectly fine at 25 degrees. You need to use these in a buffered, slightly alkaline or basic solution. No, they worked in tap water and even in seawater. And from the process, we could get our protein hydrolysate or the mineral stream. And we've mainly been focusing on the mineral stream. But the protein, what we did was we allowed that to dry or freeze dry it. And we spoke to our colleagues at the Ocean Science Centre here who do a lot of fish feed trials and things like that. And they said, oh, probably the simplest thing, because you're never going to produce enough that we could use it, is just get yourself some zebrafish and have some of them as your control, where you feed them commercial fish food from an aquarium shop, and then the others you feed them your protein hydrolysate. And we had these fish for probably a couple of years before we moved laboratories, and they were perfectly happy on their mussel protein diet. So moving away from our little fish experiment, we did a number of things with our mussel shells, and we're still working with our mussel shells. And one of the things that we discovered, and this is serendipity and how important it is in research, is that if you heat up the shell to a high temperature, the inner nacreous or shiny, lustrous part of the shell separates from the outer hard part of the shell. So this was purely done by chance. My student Jennifer was trying to make sure her mussel shells were dry for future experiments. She left them in an oven overnight and then found out that they would start to flake apart. So we got a bit intrigued by that process. And we also were intrigued by a lot of the literature talking about nacre and growing nacre, all this shiny material artificially, for a whole bunch of reasons like dentistry applications, that sort of thing. So what we also found was our bio-sourced nacre, we could use that to make synthetic nacre. So both of these are a form of calcium carbonate called aragonite. But on the left, it's just mixing the two together. Whereas on the right, we put in some seed crystals and also some of our protein hydrolysate. And you can see a very different form of calcium carbonate growing. So for a short time during her PhD, Jennifer had a little cosmetic spin-out company because we found the nacre so attractive, and she found that you could also dye the nacre because basically it's similar sort of material to eggshells with food coloring dyes, and she used it as an additive in nail polishes. And one of the final things she discovered finishing up her PhD was that if you treated the mussel shell materials with acetic acid, you could get a strange change in the morphology of the calcium carbonate. So now we're looking at the calcite. This is tightly bundled in the micrograph on the left, but after washing, treating with acid, filtering, and then washing and drying, we would get a strange nest-like material. And here's a little short video. And this is Jen very excited when she first discovered it. It's not really cinematography at its best, but you can see it has a kind of gummy-like texture. So on the right here, she's pushing down on part of this sponge-like soft calcium carbonate material that I remind you comes from a hard shell. And all we've done is wash it, treat it with diluted acetic acid, which is vinegar, and stir it around a bit, and it forms this strange material. So it's kind of like a gummy feeling when it's wet. On the left here, she's just showing you some traditional calcite from the shell and how brittle that is. And when it's dry, it's a little bit like the texture of insulation fiberglass, and it absorbs a lot of water. So Jen then went on to look at the use of the soft calcite material in supercapacitors, doing some electrochemistry experiments. So she was lucky enough to go to Monash during her PhD, and this paper was just published last year. It took a while to get that one published, and then she did a couple of postdocs, and now I'm very proud to say she's an assistant professor in the chemistry department at the University of Guelph. Moving back to the salmon, and this research that I'll talk about briefly is by Courtney Laprise, who's now a high school teacher. This was basically a collaboration with my colleague and husband, Chris Kozak, and an engineering colleague called Kelly Hallbolt. So Kelly had been working with the Newfoundland aquaculture industry for some time and had developed a process for taking their salmon waste and getting the sort of oil from that. And she tested it for use as a heating oil in fish processing plants and that sort of thing. And I'm like, you know, this is too nice an oil to just be burning. We can do something interesting with that. And, you know, obviously it could be used as a nutritional supplement, as fish oils often are, but to be used as a supplement, you need very stringent conditions and processing that maybe isn't affordable at the sites where the salmon is being processed in Newfoundland, and also because of geography and lots of other competing kind of economic factors. So this picture at the very top is meant to represent a typical fish oil molecule. So these are triglycerides mainly. It's about 70% triglycerides. And we have different fatty acids on each arm here. Obviously, in some cases, they may all be identical arms or might be quite different to this. So what we do is we use a catalyst to add oxygen to the triglyceride. Then we use a different catalyst to react our new oxygen containing material with CO2. So now we've got these blue atoms appearing in the structure. And then we take what's called an amine or a diamine, and it reacts and cross-links and forms a polymer. The amine we have reported so far, it does work with other amines, is something that's called cardylite NC540. This is actually a commercially available material that's being used in adhesives for industrial carpeting and things like that. But where it comes from is interesting, is that it's made using the waste from cashew nut shells. And when we were making this new polymer, we wanted to check it happened. And so you can see here, we've got OH groups in the bottom structure that aren't present in the top structure. So we use a technique called infrared, and the circle on the left is highlighting that OH groups are now present. And also we're seeing some changes with the two red circles on the right. So we've got a new peak appearing and a smaller peak in one position, which again is telling us that our reaction has been successful. Of course, we could also tell using our eyes that the reaction was successful. So we make these transparent red film materials. We could confirm that there was nitrogen in the materials and they were quite stretchy. So we've been lucky to get funding in the past few years since we first started this project to get some new equipment so we can actually measure the mechanical properties of the film a little bit more. And we've also been looking at some of the other properties of these material. So this is a photo of one of my PhD students, but it's actually back when she was doing her honors research with me. And she looked a little bit at degradation of this fish oil derived polymer. And she did a number of different studies. And the photo on the right is showing some fungi growing on the surface of that polymer. So we've collaborated with Clarissa Sitt, who's a bioorganic chemist or a chemical biologist at St. Mary's University in Nova Scotia. And so we send materials to her. Her students grow more of the microbes and then send them away for genetic sequencing, Genome Canada. And the fungi and bacteria growing on the surface of our polymers, these are ones that are just present naturally in rivers and in the air and that sort of thing. So it's promising that this material really would be degradable in the environment if it got out there. So during the beginning of her PhD, while starting that project, Michele presented virtually at an ACS meeting, American Chemical Society meeting, and people were quite excited by the research that she presented. And the question that the people interviewing me and Michele would always ask is, does it smell? And the answer is no, not once it's in the polymer form, it doesn't smell. So hopefully we'll be able to understand this material a little bit more in the future. And as I promised in my abstract, I'd talk about some CO2-based chemistry. I'm moving on to that. I'm not going to go into the nitty gritty, and maybe I'll have to catch up with Shrin sometime via a separate Zoom call to talk about the chemistry in a bit more detail. But Coriandria, she was the lead on this, and she wrote up during lockdown and is now at dentistry school in Dalhousie, and Michele has taken this project on as her own. So As you'll have seen, we like to react carbon dioxide with things, and particularly these three-membered ring molecules called epoxides or oxiranes. And many people use metal-containing catalysts for these reactions. But what Corrie discovered was if she used boron-containing catalysts and a range of different co-catalysts, she could get these reactions to work really, really well. So this TOF thing is a turnover frequency, and that's telling us how fast our catalyst is. And so basically what it's telling us is that one molecule of our boron could produce over 1,000 molecules of our product in 1 hour. And that's pretty fast for this sort of reaction. But what we could also do is make polymers, and we could also make what are called functional polymers, so those with, say, an amine group on it or other groups. And what we also discovered was that we could use the same catalyst in one pot to do a series of reactions. So this saves time, money, energy, and other chemicals. And so what we found we could do was make a polymer. Then if we had what's called an alkene group, a carbon-carbon double bond, we could add things to that double bond by increasing the temperature and then adding in our reagent. And we're quite excited to kind of look at this in a lot more detail moving forward. One of the things that we're also interested in is using waste and moving back to land-based biomass waste. The orange juice industry, so if you had a glass of orange juice at breakfast or any other time, when this is produced industrially, there's a lot of waste orange peels, and this is used to produce a molecule called limonene that's being used a lot as an environmentally friendly cleaner. And if we oxidize the limonene, we produce a molecule that can react with CO2 and act as a monomer in polymerizations. And we can also react it with these other molecules in blue and red that we call anhydrides. They've got like a 5-membered ring with 3 oxygens on it. And so we can do things where we make what's called a random polymer, or we can make block copolymers where we have 2 different parts to our polymer or our plastic chain. And we can also make polymers where we use our 3-membered ring epoxide, our anhydride, but then also introduce CO2. And we can look at a lot of our reactions using special instruments such as infrared spectroscopy and be able to check that our reaction is happening properly. But what Michele was key to discovering in this process was that we could actually depolymerize or chemically degrade our polymer using the right sort of catalyst. And so we have one sort of boron compound shown in blue in the top right, and that helps us make our polyester polycarbonate. Whereas if we have another sort of boron-containing compound shown at the bottom left, we can selectively degrade part of our catalyst. And so Michele's been looking at degrading polypropylene carbonate, which can then mean that we've got a cyclic propylene carbonate that can be used as a green solvent in batteries and things. And then we had a couple of undergraduates who've been following up this work over the past few summers, making renewable anhydrides. So what they've done is taken terpenes. So on the reaction scheme at the top right, there's a molecule without oxygen. This is isoprene, but there are many molecules like this that can be got in the essential oils of trees and fruits and all sorts of things. And what we found was we can take this with the other molecule shown on the left of the equation and put them in a microwave for 10 minutes, and we produce our anhydride, which is now bio-derived, because the middle molecule on the top right is called maleic anhydride, and that can be obtained from biorefineries. And the added advantage of this is that it works really well in 10 minutes. Because we get very high conversions and yields of our product, we don't have to waste time separating it very much, and it works really well. So Michele's been busy doing lots of polymerizations with different terpenes, and using lots of fancy characterization techniques to understand what is happening. And so we should have this story finished before the summer, and hopefully submit a manuscript soon. So hopefully I didn't lose you too much on those last few slides with all the chemistry structures, but hopefully you've learned a little bit of something about using biomass and CO2 as building blocks to make something new. So I started by talking about how you can disperse biochar in environmentally friendly solvents, how we can use biochar with oxygens on the surface to catalyze reactions. We can remove the protein from shells and even do reactions with enzymes in seawater, and then use that calcium carbonate in a number of applications. We can use waste fish oil and make polymers, and use different catalysts to make polymers and small molecules from CO2, and also degrade polymers. So this is a picture from last year of the students in my group, and a list of collaborators and funding agencies. This is a little iceberg, which is about a 5 to 10 minute drive from campus a few years ago. And in the background, that's a beautiful blue whale skeleton. So we moved into a new science building a couple of years ago now, and it's just a fantastic place to work. And this blue whale skeleton is just outside my office, so it acts as a real big inspiration to keep trying to do good things for the environment. So thank you for your attention. So I'll go ahead and read out Francis's question. So it says, how can policymakers, industries, and consumers contribute to the advancement and adaptation of sustainable polymers, materials made from waste, biomass, carbon dioxide? So before I came to Memorial University, for a short time, I was at a university in the UK, the University of York, and they have a Centre for Green Chemistry. And they found in some of their research and working with social scientists that you shouldn't underestimate the power of the consumer. And the consumers can really make policymakers and industries change their minds. And so they found that they were actually approached by this company called Boots in the UK. So that's like Shoppers Drug Mart in Canada or something, to try and help them understand the life cycle of the materials that they were selling, whether that was cosmetics or pharmaceuticals. And it was really the pressure of their customers saying, we want something more sustainable, that got them to do that. I think policy does have a really, and legislation has a really important role to play on convincing industries to make changes. But I'm not going to lie, it's really tough. So with, for example, plastics and polymers, where there's been so much investment over decades, if not a century, into synthetic polymers made from petrochemicals, they don't want to change. They don't want to have to invest in new infrastructure. So one of the things that people like me are trying to do is just be approachable for a start, but also to make sure that what we're doing where possible can kind of be a drop-in, slot-in replacement with existing infrastructure. But that's a really good question.
Can I ask a question then? It's kind of connected with that. So as you're developing some of these technologies, is part of the equation trying to minimize the transport of those waste products, like trying to develop technologies that can be done on site? Because I can envision some scenarios where you're not really getting ahead because you've got to
so one of the things, one of my recent PhD students, so it was a funny relationship. So there's a place called Marine Institute of Memorial University, and their director of their Center for Aquaculture and Seafood Development. She had an MSc in biochemistry, but she really wanted a PhD because administrators in the university were like, you know, you need to grow the graduate program at the Marine Institute, and to do that, you need a PhD. So I supervised her in the environmental science program, and she was looking particularly, because she already had some data from her research already there, at the crab industry. So she interviewed stakeholders, so non-structured interviews for the crab fishery, so people who were fishermen, processors, industry associations, all sorts of people. And also one of the things she did was a kind of techno-economic assessment of where perhaps would be the best location if we were going to have a crab shell biorefinery. Where should it go, and kind of what historically has been happening in that sector, and how much people would be willing to do. And I think in Newfoundland, the problem is we have a really long coastline, and a lot of these industries are scattered around everywhere. But for some of the processing, it really needs to be done at a centralized site. So most people probably don't know the geography of Newfoundland, but you know, there's like this bit sticking out into the ocean called the Avalon Peninsula, and there's some fishing, and most of the people in Newfoundland do live on the Avalon Peninsula. But a lot of the fishing industry takes part across the kind of main kind of Atlantic facing coast, but also on the northern peninsula that sticks up towards Labrador. And so they found that or one of her conclusions was that it would be good, if possible, to have two bioprocessing plants, one in central Newfoundland and one in the Avalon Peninsula somewhere. But yes, the technologies really have to meet the needs, not imagined needs, but the real needs of the industries, and also be kind of, you know, think on your feet and try and think of local solutions to local problems, but also be aware that some of, you know, the things that affect Newfoundland in terms of fishing also affect other fishing communities all around the world, in that, you know, sometimes, you know, that you're not going to have access to you know, great water supply. You know, you've got ocean water, but you haven't got a lot of clean municipal water, and that sort of thing.
Okay, so thanks, Fran, for an awesome talk. I've got a couple of general questions. So the first one would be, like, when you're dispersing the biochar, I'm sure you've considered the use of micelles as opposed to combination of organic or inorganic solvents.
Yeah, so that is a potential way of dispersing the material into micelles. And we don't know as well, when we were doing the polymerization with the caprolactone, we were doing, it was kind of like a graft polymerization, that we were attaching it to the surface, and whether that was having an effect on the material. But yeah, so in my past life, I was really interested in solvents, which is kind of what inspired this work. And so um, for many people, they were like, well, this is heterogeneous. You can't use these Camelot-Taft parameters in this way here. But we saw really good, strong correlations. I think potentially if we somehow made and designed the right sort of surfactant, that would work well for dispersing it. But again, that's adding an extra layer of complexity. So I'm of the kind of keep it simple, stupid school of trying to do green chemistry. I don't want to overcomplicate things if I can. I mean, I was chatting with a colleague earlier and I was like, oh, I think we should try this. I said, it's not going to be particularly green, but it's just interesting just to see what happens.
Yeah. So the next question I have is basically coming from somebody who's worked on aragonite and calcite for a little bit in Montreal. I was curious to see the calcium carbonate can be gummy. Like in my mind,
Yeah.
a gummy calcium carbonate would mean there's some kind of protein bound to it that's
Yes.
prohibiting
So there's about
crystallization.
like, I think less than 1% was kind of what we said of some sort of protein mixed in there. Whether it is chemically or physically attached to the surface of the crystals, we don't know, just because it's
Hmm.
so hard. It's there in such small quantities relative to the calcite needles. It's hard to measure and quantify in any way. And so when we kind of clean the mussel shells and separate the calcite from the nacre, we did solid state NMR of the calcite material, and you could see that there was protein residue there. But it was just a solid state proton NMR, and the signals are all really broad. And so you can say, oh, look, there's some aromatic residues in these amino acids within the protein, but that was kind of really about it. And yeah, it's just when she first did those experiments and there was these white blobs kind of suspended in the water, I thought somebody else in the group was playing a joke on her, that somebody had got some filter paper, screwed it up into little balls, made it gummy and thrown it in there. So that's where the video comes from. She's like, no, this is real. This is so she kind of sent me that in a message once. Yeah, and then we just tried repeating it a few times. Yeah.
Cool. And the last question is probably better to deal with it later. I was blown away that you can use a more Lewis acidic boron uh, to depolymerize
Yeah.
and use a less Lewis acidic boron to do the
Yeah.
polymerization side. That was fantastic. But yeah, we can that's more, as you said, the
Yeah.
nitty gritty of chemistry, so we can talk about that later.
If you're willing to field one more question, Fran, there is one from the chat. I'm not sure if you can see it, but Carson has a question kind of related to the previous one.
Yes. So it said, yeah, the KPI, could a material like this have any consumer industrial uses, or was it just a really cool finding? So in the interest of time, I kind of cut out some of the stuff that we tested that gummy material on. So one of the things we did was we dried it out and then we tested it, and we saw that it could take up 20% by weight water. And so that was interesting. So then we were trying to see, oh, could we trap water from the air, or could we do other things? And it wouldn't take water from the air and that sort of thing. So that was a dead end. So then where it was taking up water and absorbing kind of that and becoming this gummy texture, we tried adsorbing dye. So it works okay for absorbing dye, but there are a lot better materials out there for remediation of water that's been contaminated by dye. But what it was quite useful for, but this is not amenable probably to a large scale setting, because of just the amount they would need, is that we found that it could absorb oil onto its surface. So, and it could absorb quite a lot by mass, like 1,000 weight% by mass. And you could then clean the oil off and reuse it. So this is crude oil. So I got a sample off somebody here who's collaborating with the oil industry, and we made some pretend seawater using salts. So you can get at aquarium shops, you can buy marine salts. They're much cheaper than chemical suppliers of salts for making fake seawater. And we put our sponge in there, and then we made an oil slick, and it would take up all the oil, and then we would use a tea strainer, very high tech, to remove our sponge material, and then we would clean it, and then we could reuse it, and it worked quite well. But this sponge material, when we're making it, is only a small amount of the mass of the shell. So when we're treating with acetic acid, we produce a lot of calcium carbonate. So the aragonite part of the shell all reacts, and some of the calcium carbonate, and we're left with the sponge material. And so in terms of scale up, it's not going to work. So the sponge material, we're really interested, just because it's more separated needles of calcium carbonate, in terms of incorporating it into other materials. So one student's been looking at drug delivery materials, so kind of very high value areas, because you wouldn't need as much of that material then to make a difference. And then we've also been looking at it in energy storage materials to sort of just reduce the amount of some of the more environmentally harmful things that are present in batteries and supercapacitors. So for example, there's a lot of fluorinated polymers in there. If we put in 10% of our gummy mussel material mixed in somehow, does it reduce the efficiency? And the answer is no, it doesn't really reduce the efficiency of that material or the strength of the membrane or whatever we're making. But there's still a lot more work to do. But it is, you know, I just find it a really cool finding because it was just so unexpected.
I have one quick question though. In Manitoba, it serves that we should go to an aquarium store to get seawater, but why would you in Newfoundland? You could just run down to the beach.
Yeah, so we would also, we do go to the beach and get it in a bottle and kind of run away from the cold water and things. But why we go to the aquarium store is just to make sure we have saves us sterilizing it, basically, that we've got no algae and other things in there, and that we can report accurately without having to do the measurements and experiments, what the different ion concentrations are and what the conductivity is. What's really cool at the Royal University is we have two places. So we have the Marine Institute, we have our main campus in St. John's, and then we also have the Ocean Sciences Centre. And at the Ocean Sciences Centre, that's in a place called Middle Cove, or near Middle Cove, they actually have seawater on tap. So they have pumps that go down into the ocean and pump seawater in. So yeah, you know, if I really, and I think in their cases, they probably have some parts where they have filters to remove the algae and microbes and things, and probably ozonolysis and things like that. But yeah, if we needed to scale something up in seawater, we could probably speak to the people at the Marine Institute. Yeah.
Good point. Carson says, thank you. Yeah, it's a very interesting question and interesting answer. It is past 3:30 and I know it's much later where you are as well. So I'm not seeing other questions. So I'd like to take this time to thank you for taking time to join us on Zoom for a really interesting talk. Yeah, I know it's late on a Friday, so we really appreciate yeah, a few thank yous in the chat as well. So thanks again, much appreciated.
Thank you. My pleasure.
All right.
Hopefully I'll make it out to Brandon at some point in the future and see you all in person.